was the DNA, but it was at a very low concentration. So what you wanted to do was to amplify all this DNA so you had enough so that you could actually cut it successfully and be able to visualize it. But the very first thing you need to do to amplify it is you have to make, well, you have to obviously make more of it, so how are you going to make more of it? You're going to take the DNA and you're going to put it into bacteria, and that step's called a transformation. That's the very first thing you did. Right, so the very first thing you did is that you will get your bacteria, which are DH5 alpha competent cells, spell, or 2. And with these cells, you would add your DNA. Now, if I recall properly, we gave you 100 microliters of the bacteria, and you added 2 microliters of your unknown DNA. And then, how long you let it sit for? Like, is it 10 minutes? Less? Okay, no one knows. Okay, so I know you all have it saved, so you're going to have to look it up. So what you did next was sit it on ice. Alright? So this step is actually a very important step because what happens here is that you're going to have your bacteria cells lying in your solution as so, with these little DNA particulates all over the place. What happens here is that, so picture this as as the membrane of the cell, and the DNA is going to sit right on the surface. The longer you let these bacteria sit on ice, the more these plasmids get to lay on the surface. So they get there, they get embedded right into the membrane within the sugars and such. And then when you do the heat shock, which was the very next step, 42 degrees Celsius for 90 seconds, I believe. This heat shock would actually create holes in the membrane, which would allow DNA to go through. So now, with this heat shock, you're going to produce your bacteria, which have your plasmids in it. Okay. After that, you would immediately put the cells